let's look at some stress testing data from 15 compounds. So because of the questions around, I won't tell you all the background, but the questions around genotoxic mutagenic impurities, there was interest to know how accurate we were in our stress testing. Were we going crazy in producing way too many degradation products, or were we reasonable? So we looked at stress testing results from 15 compounds that we had marketed formulations. So that means it made it to phase 3 or onto the market. So we looked at the API and the drug product stress testing results. That means we looked at the API and all those acid, base, oxidative, and we looked at the drug product under accelerated heat and humidity. And I'll show you those conditions in a slide. And we looked at both the full accelerated and long-term stability results with and without optimum packaging. So that's a pretty complete data set around all these compounds. And here's the stress conditions that over the years represent essentially the way when I was at Lilly, the way we settled in on and conducted stress testing. So for hydrolytic acid base, we would look at a minimum pH 1, 7, and 13. Sometimes we would do it in increments of two pH units to give us more data, but at a minimum 1, 7, and 13, up to 70 degrees C for up to 1 week. That's about as long as our experience would say you need to go. The solid state, we don't, I advise 70 degrees C and 75% relative humidity. So you get a high and low humidity, and 2 or 3 weeks. 3 weeks is probably as long as you need to go, but both the drug product and the API. For photolytic, the stress for photostability is, I recommend 2 to 5 times the ICH confirmatory guideline, and we always use option 1, which is a little bit more rigorous exposure. It's simulating sun versus indoor lighting, which is a more complete exposure. And we would do both the solid state and in solution for the API, and just the solid state unless it was a parenteral. And for oxidative, this is where most companies that I run into have holes in their procedures. Most people do a peroxide stress, and that's something like 0.3% hydrogen peroxide, room temperature 1 to 3 days. We would only look at that for the API. But I recommend that you're leaving big holes in your study if you don't use a radical initiator and if you don't do transition metals. But the two most important ones are peroxides and radical initiation. We conducted all of these studies was to stress it to the endpoint regardless of whether there was degradation observed. So if you didn't see any significant degradation at the end of those time points and conditions, it's stable to acid or it's stable to whatever. And we would define major. We would focus in on the compounds that were produced as major. So you can get a lot of grass, a lot of small peaks, and if you try to focus in on all of those, then you really have an explosion. So we really think it's best to focus in on the major ones, and you can have an algorithm for defining them. And we did that by 10% or more of the total degradation. So that is, if you have an API that lost 20%, 10% or more of that would be 2%, right? And then you have to, you only focus in on the peaks that are at least 25% of the largest individuals. So if you had 10% degradation and you had 1 peak at 8% and 2 peaks at 1%, well, those 1% peaks are 10% of the total degradation, 1% divided by 10 times 100. But they're not 25% of 8. So you can kind of get rid of some of, that's how you keep the stress studies from exploding and get you focusing in on the ones that are most likely to be realistic. And this has been documented in a couple of publications, and I include them here if you want to look for more detail. So looking at those 15 compounds, here's what it looked like. From our knowledge space was 124 total compounds out of 15 products. And so it's not exorbitant. It's about 8.2 major degradants per drug and drug product. So it's not an excessive number. Some people would think, oh, you must be looking at 30. Well, if you're looking at the grass, the very small degradation products, maybe so, but if you focus in on the major, it's not that many. And if you look then, so that's our knowledge space. Our design space, if you want to call it, would be what can form in that drug product under the long-term storage condition or the accelerated storage condition, but not really packaged as we're finally going to have it. And that went from 124 to 45. And then what does actually form in the final drug product, when you package it well, it's 39. Now it looks like 45 to 39 is not a very big decrease. So it tells you that the accelerated is pretty predictive, but the levels of those 39 are coming down substantially. So we might not have had 39 had we not known there were 45. By that I mean they're below maybe what we would have identified, but now that we know that that peak was there, we can say, oh yeah, at 0.06% there's that peak, at 0.03% there's that peak. So, but it gives you an idea that that kind of stress procedure is pretty good. Now, I'm not trying to claim that it always is inclusive of everything like this was. It was a perfect subset, but our procedures at Lilly evolved. I was responsible for stress testing for my entire career, so they evolved over the years because I got caught with surprises, and that's how we ended up developing these procedures so that we wouldn't anymore. Getting caught in late phase development is very expensive and embarrassing and job threatening potentially. So my assertion is that what we want is we want our stress testing studies to have more degradation products than long-term. We don't want it to just produce what's going to happen because we want confidence that our method will detect all of the long-term deg. We want it to be a subset so that we know that we're comprehensive. Why do we want to reveal all of them? It's difficult to anticipate which of those major degradation products that you observe in stress testing are going to be the ones that are relevant, and you need a method that can detect it.